Hello, everyone. This is the video for the Life Sciences practice test, questions 16 through 20. So let's start with question 16. For which of the following reasons is it better to clone cDNA rather than genomic DNA into bacteria if the bacteria are to synthesize a eukaryotic protein of the correct sequence? So A, the discrepancies between the genetic code of prokaryotes and eukaryotes. There are discrepancies. B, transcription factors for prokaryotes and eukaryotes differ. C, bacteria cannot process mRNA to remove introns. Or D, bacteria cannot modify proteins in the same way that eukaryotes can. So first, let's define cDNA versus genomic DNA, because that's really what they're asking here. So genomic DNA is all of the DNA in an organism. cDNA is only the DNA that's expressed. So meaning that DNA is transcribed to mRNA, and then in this case, back to DNA. So basically, if it's not being expressed, we're not talking about that part of the DNA. So introns and extra stuff that's cut out when it's made into mRNA, that's not expressed. So therefore, that doesn't get transcribed and then therefore get back in DNA. So basically, we're looking here, cDNA in this case is basically going to be DNA that doesn't include extra stuff like introns and things that don't get expressed. So let's go through the wrong answers. So A here is incorrect. A says there are discrepancies between the genetic code of prokaryotes and eukaryotes. This is incorrect because prokaryotes and eukaryotes have the same type of genetic code. There's no discrepancies. They're both made of A's, T's, and G's, C's, all the same. B is also incorrect. B says the transcription factors of prokaryotes and eukaryotes differ. So let's define transcription factors. Those are that's a protein that controls gene expression by binding the DNA and regulating the rate at which those are going to be transcribed. So the key here is the transcription factors have to do with rate, and we're not looking at rate. Excuse me, rate is irrelevant to this question that they're asking. So B is also incorrect. D is incorrect because it refers to protein modification. D says bacteria cannot modify proteins the same way that eukaryotes can. So again, incorrect, it's because it's referring to protein modification, but the difference between cDNA and genomic DNA is the lack or presence of introns, which is dealt with in transcription and translation. We have to get past transcription first, which is what C talks about and why C is the correct answer here. So C is going to be the correct answer because bacteria cannot remove introns. It's a big thing with bacteria. They don't have introns, they can't remove introns. So if we're going to introduce our DNA to bacteria, we have to give them DNA without those introns. So genomic DNA is going to have those introns, but cDNA is not going to have those introns, making it the correct answer here. So the biggest thing with this question is knowing the difference between cDNA and genomic DNA. That's really what they're hitting at here. All right, question 17. So they start out by giving us these percentages here. This question is going to deal with dihybrid crosses right off the bat. You can see here we've got two different traits we're looking at. We're looking at R and T, and they've given us a percentage breakdown. So let's read the question. So question 17 says, in a particular species of fly, gene R can be found in two allelic forms, capital R, which is dominant, and lowercase r, which is recessive. Similarly, gene T can be found in two allelic forms, capital T, which is dominant, and lowercase t, which is recessive. A female fly of a similar genotype, capital R, capital T, little t, basically heterozygous for both traits, is crossed with a male genotype fly of little r, little t. So homozygous recessive for both traits. The progeny are examined with respect to the genes R and T, and their genotypes are determined. The results are shown in the table above. Which of the following is most consistent with the data? So A, gene R and T are on different chromosomes that exhibit independent assortment. Gene R and T are linked on a single chromosome. C, gene R and T are both on the X chromosome. Gene R and T, sorry, then D, gene R and T are located on different chromosomes that do not assort independently. So there's a lot to break down in this question. So the first part of this question is referring to, this is all about meiosis. So the first thing that's important is to understand those steps of meiosis. So I'm going to skip this in terms of review because it's not super imperative to this question. However, I'm going to post a link here for you guys. That's a good video. If you've ever seen Amoeba Sisters, they do a really good job of breaking it down into simple terms. So I'm going to post a link here that you guys can see this meiosis overview video that does a really good job of going through some big vocabulary on meiosis. It also really goes through all of the steps of meiosis 1 and meiosis 2. So if you need a review on that, I would highly recommend going and looking at this first link. So again, I'm going to kind of somewhat skip over that. But again, good review. If you feel like you need a review over that, please go watch that video. So the next part of this question that we really need to look at is this idea of the law of independent assortment and linked versus unlinked genes. So for that, I'm going to go to this second link here. Again, you can see it here if you want to write it down. I'm going to go ahead and pull up this link. So this link is also a really good resource. It also goes through what happens in each of the steps of meiosis 1 and meiosis 2. You can see descriptions it's got here. It's got a really good video, or sorry, not video, a really good diagram so you can see what's going on in each phase. Talks about crossing over, homologous chromosomes, a bunch of vocabulary. And homologous chromosomes is going to be really important here. So basically, homologous chromosomes, we number our chromosomes 1 through 23. So in meiosis, you'll have chromosome 1 from mom, you'll have chromosome 1 from dad, chromosome 2 from mom, 2 from dad. Those are going to be homologous and they're going to have the same genes on each chromosome. So let's just say chromosome 1 has gene A for mom, chromosome 1 from dad will also have gene A for mom. And those are going to line up right next to each other in meiosis. So that way, when we separate, you always get 2 copies. You always end up with the same amount. So here, I want to, this law of independent assortment right here, this is what we're going to harp on here. That's really important for this question. So it says the separation of alleles for one gene will occur independently of the separation of alleles for another gene. So basically, we just said there, I gave the example of gene A. So let's say gene A is on chromosome 1. Gene B is on chromosome 2. What this is saying is that gene A that's on chromosome 1 is going to separate to a particular cell completely independently of gene B that's on chromosome 2. Those are not going to have any sort of linkage. They're going to randomly assort. They could end up in cell 1, cell 2, cell 3. You'll see here at the end you end up with 4 cells. So gene A for mom, for example, could end up in any 4 of these cells. Gene B could end up in any 4 of those cells. So they're not linked. You're not going to get both A and the same A and B here in this cell, and then a different one in this cell. It's all random. And that's because here's a diagram that I think is really helpful. So you can see here we have gene A on dad and mom, and we'll just call this chromosome 1. So you can see, in fact, I'm going to annotate here. So we can see gene A is on chromosome 1. I'm just going to put a 1 here. We're going to call this chromosome 1 from mom and dad. This is chromosome 1 from dad, chromosome 1 from mom. And then down here we have chromosome 2. You can see that that has gene D. D from mom, D from dad. So the law of independent assortment says that this A on chromosome 1 is going to go to a random cell completely independent, in no way linked to this D. So you could get this A from dad, and you could get either this D, little d from mom, or you could get the big D from dad. It's random. It could be either one. Those are going to assort independently of each other. As opposed to B here is on the same chromosome. So if I get chromosome A from mom, I'm more likely that I'm also going to, or sorry, from dad, I'm also more likely to get chromosome B from dad. Those are on the same chromosome. Those are linked. They're likely to go together. It's possible that they might not, based on crossing over, depending on where it crosses over. But basically, if they're on the same gene, they're linked chromosomes. They're more likely to go together as opposed to being on different chromosomes. There's no attachment. It's completely random. They're going to end up in different cells, potentially. So you can see here all the different combinations. So now let's go back and look at our question here. Is that my annotation? Here's our. All right. So right off the bat, based on talking about that law of independent assortment that we just went through, we can eliminate answer choice D. Answer choice D says genes R and T are on different chromosomes that do not assort independently. That will never happen. If they're on different chromosomes, they're always going to assort independently. So if we go back here, A and D being on different chromosomes, chromosome 1 and 2 are never going to be more likely to go together than chromosome 1 and 4 or 1 and 5. It's always random. So if they're on different chromosomes, they're always going to assort independently. So that means we can eliminate answer choice D. Right off the bat, D is gone. So now kind of the third part that we have to go through with this question, now that we've looked at independent assortment and linked genes, is we need to see if those apply in this question. So you can see the other answer choices have to do with being linked or being independent assortment. So we need to figure out if the data they're giving us here supports that these two genes are linked or whether they're not linked. So that leads us to our third focus of this question, which is going to be Punnett squares. So in the case of this question, we're going to need to use a dihybrid Punnett square because we have two different alleles. We have capital T and we have lowercase t. So there are two ways of doing a dihybrid cross. I'm going to post a video to the most traditional way. So if you've kind of forgotten how dihybrid crosses work, go back to this. This third link here, traditional dihybrid cross video. This is another Amoeba Sisters video, but they do a really good job of walking you through and explaining. Oh, there's the eraser. Of explaining dihybrid crosses, how they work, things like that. So again, I'm not going to pull that up. I'm going to do what I call a simplified dihybrid cross, whichever way you remember or you were taught works fine. You're going to get the exact same data. Oh, so how do I get back to? So I'm going to stop sharing my screen. No, I want to keep sharing my screen. How do I go to? Let's see here. I'm trying to get over to, well, we'll just scroll down and we'll just do it right here. Just annotate in the space. All right. So we are going to talk about dihybrid crosses here. So again, I'm going to do it this what I call the simplified dihybrid crossway, which is basically we're going to do two separate typical Punnett squares, and then basically you multiply them together to get your percentages. But again, you can do either method, but the idea here is that we need to figure out, we need to do a dihybrid cross, and if the percentage we get with that dihybrid cross, if it matches what's in there, what the data they give us, that means that those genes are independently assorting. That's how that anytime you do a dihybrid cross, those are going to be for genes that are assorting independently. So if they're sorting independently, our data is going to match the data that they get in their question or the data that they give us. If it does not match, then it means they don't sort independently, which means we can eliminate A and we're going to be left with B or C. So we're going to start out with, let's do our Punnett squares here. All right, so again, I'm going to do 2, is what I call the simplified dihybrid cross. And basically, I'm going to cross gene R on one Punnett square and gene T on the other. And then we can multiply them together. So, go back to our question here real quick. So we are crossing, I'm going to write what we're crossing over here. We are crossing a fly that is capital R, capital T, little t, with one that is homozygous recessive for both. All right, so basically what we're going to do is we're going to do the R's on one Punnett square and the T's on another. So I've got parent 1, capital R, and I have parent 2, little r, little r. So I'm going to do my Punnett square, put them both together. Pull the big R, pull the little r, pull the little r, pull the little r. Big R, little r. All right. So now let's figure out all of our phenotypes here. So we have, in this case, we're going to have big R, little r is going to be 50%, right? And then little r is going to be 50%. So now we're going to do the same thing over here with our T's. And in this case, sorry, on this R1, in this case, we have none that are capital R, capital R. I just shouldn't write that down, but we have 0. So now we're going to do the T's. So we're going to do the same thing. Big T, cross with little t. And again, pull the top down and the side across. Perfect. So again, we're going to write our percentages. Let's do our heterozygous first. Again, we have no homozygous dominant, so I'm just not even writing it down, but we have 50%. We have 50% heterozygous, and then we have 50% homozygous recessive. So now we need to figure out what percentage we need to basically combine these. So if I want to know what percent are going to be homozygous recessive for both, we multiply the heterozygous for the R's and the heterozygous for the T's. So if we multiply 50% by 50%, that gives us 25%. So kind of running out of space here. So we're going to write capital R, capital T, little t. That should be 25%. And then if we want homozygous recessive, or sorry, heterozygous for R, homozygous recessive for T, again, we're going to multiply those together. So we would have big R, little t, multiply 50 by 50, and we get 25% again. It's hard to write on this. So then that takes care of our homozygous or heterozygous for R. Now let's do our homozygous recessive for the R's. So we have little r. We're going to combine with big T, little t. So again, we're going to do 50% times 50%. It's going to be 25%. And then our last percentage here, little r, big little t. So homozygous recessive for both. Again, we're going to multiply homozygous recessive R, homozygous recessive T, and that will give us 25%. So basically we have 25% across the board for all of our percentages. So now let's go back to the question. And I'm going to leave that up there, that annotation stuff. It's going to overlap our thing here. So I'll scroll down a little bit. So the data we get from them says that heterozygous for R, heterozygous T is 39%. As we can see here, that does not match, right here, that does not match our 25%. Neither does, none of this data matches. Capital R, heterozygous for R, homozygous recessive for T, 25 versus 11, 25, 25, 13, 39. So again, none of that matches. So I'm going to scroll down again. I'm going to leave this up here just in case anyone's writing it down. So the fact that that our data does not match theirs tells us that we can eliminate answer choice A. Answer choice A says they exhibit independent assortment. So again, Punnett squares and dihybrid crosses, are genes that are unlinked and follow independent assortment. So since our dihybrid cross data did not match, that means the genes that we're looking at are not assorting independently. So we can get rid of A. So we've gotten rid of A. We've gotten rid of, back here, and just cross these out so don't forget, we've gotten rid of A. We've gotten rid of D. So now we're left with B or C. So B says genes R and T are on a single chromosome. C says gene R and T are both on the X chromosome. So again, both of these are saying that they're on a single chromosome. Well, I guess if you have a girl, it could be one on each X. But the idea here is nowhere in our question, scroll up, does it ever talk about how these percentages that we got relate to male or female. It doesn't say that a certain percent of the heterozygous for both were male and a certain percent were female. So the fact that it doesn't reference the male and female in our data means that there's no indication here that it's on the X chromosome. If they gave us percentages for male and females, that would potentially indicate that it's probably on the X chromosome or the Y chromosome. But in this case, because it doesn't reference that, we can eliminate C. Genes R and T are both on the X chromosome. So that leaves us with B. Genes R and T are linked on a single chromosome. And I'm going to erase all of this now and then have us go back to that other website we were looking at, just so we can kind of confirm. Let's erase all of this. All right. So again, if we go back to that website we were looking at before, we talked about how A and B being on the same chromosome here are linked. So that's basically what they're saying. The data that we got does not support independent assortment, as we just proved. So that means they have to be on the same chromosome. So our answer choice here is going to be B. Genes R and T are linked on a single chromosome. So again, a lot to digest there. If you need help with any of that, I would highly encourage you to go back and look at one of these links. This first one goes over meiosis and all the different stages of meiosis. It's a video. The second one I think is really good. It's that website that we looked at. It goes through the different parts of meiosis and mitosis, or meiosis 1 and meiosis 2. It goes through all this good vocabulary, goes through the law of independent assortment. It gives you pictures of independent assortment and linked genes. Really good website. And then the last one goes over dihybrid crosses. So again, if you like the one big 16 square Punnett square, that works just as well as the little two small ones that I did. You can do it either way. But this link here will go through and refresh your memory on how to do the big 16 one there. All right, so let's move on now to question 18. I just realized I need to retitle this. This is question 18. This is actually question 17. So all of those go to question 17. So question 18 says, Siamese cats are white at birth, but as they mature, they become tan with much darker extremities, including the ears, nose, legs, and tail. Which of the following is a most likely explanation of the color pattern observed in these adult Siamese cats? So A, the allele of a gene required for pigment synthesis encodes a temperature-sensitive enzyme. B, an allele of a gene required for pigment synthesis is triggered by sex hormones. C, a gene required for pigment synthesis is located in mitochondrial DNA that is only expressed in the cells of the extremities. Or D, a gene required for pigment synthesis in cells is activated by increased light exposure. So the key point here that they're harping on is it's in the extremities. So why is the extremities getting darker but not the whole cat? So we're going to go through and let's look at the wrong answers first. So in this case, B is wrong. So B says an allele for a gene required for pigmentation is triggered by sex hormones. So while sex hormones can elicit change, they would not elicit change in only the extremities. C is also wrong. C says a gene required for pigment synthesis is located in mitochondrial DNA and is only expressed in cells of the extremities. So C is wrong because the mitochondrial DNA has nothing to do with pigmentation. And then lastly, D is wrong here. So D says a gene required for pigmentation synthesis in cells is activated by increased light exposure. So D is also wrong. The entire animal is going to be exposed to the same amount of light. So the light exposure is not going to be more or less on the extremities. So it's going to apply to the whole cat in this case. But in our case, only the extremities are getting darker. So if the entire cat got darker, you could make a case that maybe it had to do with light exposure. But the extremities, they're not going to get more light than other parts. So that leaves us with A being the correct answer. A says an allele of a gene required for pigmentation or pigment synthesis codes a temperature-sensitive enzyme. So A is correct because it's the only one that would explain extremities. There's a temperature difference in the extremities versus your core body. So this is why, for example, when you go outside, let's say you went outside without clothes on, your hands and feet are going to get cold first. Or this is why if you are, for example, if you go to bed at night and you pull the blanket over your head, if you're sleeping without the blanket over your head, you're not necessarily going to be that hot. Maybe you're cold, and then you pull the blanket over your head, and then before you know it, you're hot. So the core is going to be different in the temperature than your extremities will be. All right, so let's move on to question 19. So 19 says, scientists have observed that when two species of stickleback fish occur in a single lake, they have different morphologies and niches. The species differ in size and shape and in the number and length of the gill rakers that are involved in food particle retention. One species feeds in open water and the other feeds at the lake bottom. When only one species of stickleback inhabits a lake, the species exhibit intermediate morphology and niche. This scenario illustrates which of the following phenomena. So for this question, it's pretty much a straight up memorization question. You just have to know what all of these terms are. So let's go through and define them all. So answer choice A, and I'm not even sure how you pronounce that, Mullerian mimicry. So that is defined as a natural phenomenon where multiple species with effective defenses share similar appearance or warning signals. B, character displacement. So that's defined as an evolutionary process that occurs when similar species develop different traits to reduce competition for resources or reproduction. C, facilitation. A positive interaction between species where at least one species benefits and no species are harmed. Or D, competitive exclusion, which is defined as a proposition that two species which compete for the same limited resource cannot coexist at constant population values. Basically saying that if two species exist in the same environment with the same limited resource, they're not going to both be able to survive. So based on these definitions, B is the one that applies to our scenario above, just character displacement. And that's, again, defined an evolutionary process that occurs when similar species develop different traits to reduce competition for resource production. So instead of keeping their same traits and having to compete, they evolve slightly differently. That way they can kind of each exist in their niche. So species develop, so that once they kind of develop their differences, they no longer need to compete, and they can adapt for their specific sort of parts of the environment. So, which is basically what they're saying here. When they exist together, these two fish develop differences and they feed in different locations, open water versus the bottom. But when there's only one, they develop intermediate morphology and niches to kind of be able to access all of the resources available to them. All right, so last question here, question 20. Which of the following scenarios best illustrates disruptive selection? So A, female willowbirds preferentially mate with male willowbirds with longer tails. B, water boatmen occur in 3 distinct shades of brown. When a population includes all 3 shades, predatory fish eat a disproportionately large number of the most common form and reduce its frequency and population. C, guppies that are preyed on by fish that prefer large mature guppies reproduce at a smaller rate and a younger age than the more rapidly growing guppies that are preyed on by fish that prefer small juvenile guppies. Or D, under drought conditions, a population of cactus finches include individuals with short, deep bills and others with long, shallow bills, but few bills of intermediate shape that are common with normal rainfall. So they're asking about this term disruptive selection. So I'm going to go back to pull up this diagram here. Zoom out a little bit so we can see it all. There we go. Okay. So basically, these are a normal distribution of animals is going to be your bell curve or whatever you're talking about. Typically you have a lot kind of intermediate and a few of the extremes. So then there are 3 types of of selection here that we tend to see. One, it's called stabilizing selection, where for whatever reason we tend to extreme favor the middles with very few of the outside. You can see that the outsides have decreased and the middle has increased. So typically a good example of this is baby weight. I hear that one used a lot. The idea that it is really advantageous for babies to not be too big. They can't get out of mom very easily, not be too small, then they're not developed very well. They don't have enough nutrition. We really want to see baby weights here in the middle. Directional stabilization is where we're going to favor one extreme. So maybe in a particular environment, it's better for the bigger animals are going to survive more than the smaller animals. So you see a shift here in a particular direction. So in this case that they've got down here with the rats, the darker color rat, for whatever reason, maybe they live in a dark environment. And so the darker ones blend in more. And then the last one we have is disruptive selection, which is what our question is asking about, which is where both extremes are favored and not very much in the middle is favored. So, or there's not very much in the middle. So in the case of our rats, the light rats are high, the dark rats are high, but there's not a whole lot in the middle. So let's go back to our question now. So we want to know which of these is an example of disruptive selection. So let's go through all the answer choices and decide what type of selection it is. And some of them may not be on here. For example, I think the first one, so A, female willowbirds preferentially mate with male willowbirds with longer tails. This is actually not really an example of a type of selection we see above. This is actually an example of sexual selection, meaning a mechanism of evolution where organisms are going to compete for mates, resources, and food, but the idea that there's a sexual preference of these particular animals. So this is an example of sexual selection is A. So A is not going to be correct here. So next answer is B. Water boatmen occur in 3 distinct shades of brown. When a population includes all 3 shades, predatory fish eat a disproportionately large number of the most common form and reduce its frequency in the population. Again, this isn't really a true example of any of these above. This is more frequency dependent selection, meaning that they're going to eat what's most common, one, because they run into it the most, and 2, it helps to keep the ratios in check. So the higher something there is, the more likely it is to be eaten. Again, they're going to run into it more, so they're going to eat it more. So if you can eat a big fish or a small fish, for example, you're probably going to take the big fish. It's going to give you more food, more. It's just how it's going to be. So if there's a higher proportion of big fish, they're going to get eaten more. Answer choice C says guppies that are preyed on by fish that prefer large, mature guppies reproduce at a smaller size in a younger age and more rapidly, or than the more rapidly growing guppies that they're preyed on, that are preyed on by fish that prefer small juvenile guppies. This is going to be an example of directional selection because this scenario is illustrating selection in which there is a shift under these predatory conditions towards more rapidly maturing guppies. So we're shifting in that direction of more rapidly maturing guppies as opposed to the other extreme. So it's shifting in one particular direction. So that's going to be directional selection. So it's not going to be C. So that leaves us with D. Under drought conditions, a population of cactus finches includes individuals with short, deep beaks and others with long, shallow beaks, but few with beaks of intermediate shape that are common with normal rainfall. So this is going to be the correct example because it's got a reduction of the intermediate shape and an increase in the extremes, which is the definition here of disruptional selection, as you can see in that picture. So if we go back to that picture, let me get myself back, here we go. So we're favoring our short, deep bills and our long, shallow bills, but not our intermediate bills here. So that's going to be an example of disruptional selection. So that's going to make D our correct answer. So that's the video, the conclusion of the video for questions 16 through 20. Hope this was helpful. And again, feel free to go back and rewind and find those links or look at those pictures for any additional help.